Hello, scientists. Today we're going to be looking at the second half of chapter 20, which is all about oxidative phosphorylation. I want to remind you of our objectives. We actually only covered the first objective, describe the purpose of the electron transport chain and ATP synthase. We didn't even cover that. Their substances, products, and their cellular localization and tissue distribution in the first half of chapter 20. Now I want you to be able to understand how ATP synthase works, explain how electron transport and ATP synthase are functionally coupled, and how the process of oxidative phosphorylation is influenced by the availability of oxygen and NADH. What I want you to see is that there is a connection between the electron transport chain that we learned about in the first part of chapter 20 and phosphorylation that we're going to learn about now. So there were a series of energy-releasing oxidations, specifically in complex 1, 3, and 4, that gave rise to proton pumping, and this caused a pH gradient across the inner mitochondrial membrane. The differences in concentration of the ions on either side of the membrane is going to generate a voltage gradient because hydrogen ion is positively charged. So there's going to be a difference in charge across that membrane. And there's a coupling process that converts the electrochemical potential that's generated across that membrane to the chemical energy of ATP. And that's called oxidative phosphorylation. The coupling factor that does this is ATP synthase. And it's a complex protein oligomer, and it's actually separate from the electron transport chain complexes. So I don't want you to think that ATP synthase is a component of the electron transport chain. It's just coupled to the electron transport chain. However, there are a bunch of uncouplers that are able to be used to inhibit the phosphorylation of ADP without affecting the electron transport chain. Okay, so this is the model that scientists have proposed for the structure of ATP synthase. And it has these two subcomponents, the F1 and the F0. The F0 acts like a rotor and it's embedded in the membrane. And basically what it does is as hydrogen ions come in, it turns and then releases the hydrogen ions. And it's just turning as it's binding to the hydrogen ions and then releasing the hydrogen ions on the other side of the membrane. And as it's turning, it's going to turn the F1 subunit. The F1 subunit is going to act to basically take ADP and inorganic phosphate and force them together. And it's kind of difficult to understand if I just explain it. So I actually have an extra video that I'm going to share that describes how ATP synthase works. Basically, this molecule couples the electrochemical gradient of hydrogen ions to the phosphorylation of ADP. But I've also talked about uncoupling proteins or uncouplers. These are some examples of uncouplers. One, 2,4-dinitrophenol or DNP was used as a weight loss drug in the 80s and 90s, but it actually turned out to be pretty dangerous. What it does is it kind of creates a pore in the inner mitochondrial membrane so that the electron transport chain still proceeds, oxygen is still reduced to water, there is an electrical chemical gradient that's generated, but the pore that's introduced there just allows that electrochemical gradient to dissipate. And because that, there's no ATP synthesis. And this causes some weight loss because you're not making enough ATP. So you have to metabolize tissues faster to make more ATP than the uncoupler can uncouple from the electron transport chain. But also it can generate a large amount of heat that can be pretty dangerous. There's a few other uncoupling proteins. A lot of antibiotics act as uncoupling proteins as well. Okay, so I want you to know this term, the PO ratio. What that is the number of moles of inorganic phosphate consumed in phosphorylation to the number of moles of oxygen atoms consumed in oxidation. So the phosphorylation reaction we're talking about here is ADP plus an inorganic phosphate is going to produce ATP through a condensation reaction. That's why we have water. When I say oxidation here, this is actually not oxidation. Obviously, you see oxygen is reduced to water. But it's when this reaction is occurring, it's occurring because you're oxidizing in either NADH or FADH2. Those are the two molecules that are oxidized. And when you look here, you see that there's a much higher ratio of oxygen atoms consumed with NADH than with FADH2. And we saw that before, but basically what you're seeing is that you're going to produce more phosphates when NADH is oxidized than when FADH2 is oxidized. Okay, in summary, basically I want you to understand that the coupling of electron transport to oxidative phosphorylation requires this multi-subunit membrane-bound enzyme called ATP synthase. And basically this enzyme has a channel for protons to flow from the inner membrane space into the mitochondrial matrix, and then the proton flow is coupled to ATP production in a process that appears to involve a conformational change of the enzyme. There's a lot of scientific research, and your chapter even goes on to describe some of that that has been done to help demonstrate this, but it's still kind of just a model for how this process works. I want to go and explain the mechanism of coupling and oxidative phosphorylation a little bit better. We have this process called chemiosmotic coupling that's based on the fact that a proton gradient exists between the inner membrane space and the mitochondria and the matrix of the mitochondria. So why does that exist? Well, first of all, it's because the proteins that are going to be electron carriers in the electron transport chain, they're all oriented in one direction with respect to both sides of that mitochondrial membrane. They're not random, they're all oriented in a certain way. And what happens is, as these proteins take up protons from the matrix while they're being reduced, they then release them into the inner membrane space when they're reoxidized. So you can see they're going to pull them from the matrix, but because of their orientation, they're going to release them into the intermembrane space. So we're going to have a lot more protons on the inner membrane space side than we are on the matrix side. So the other thing you should note is all the reactions of NADH, coenzyme Q, and oxygen all require protons. So therefore we have to get them from somewhere. Well, where are we getting those protons from? We're getting them from the matrix of the mitochondria. So we're pulling them in from the matrix, but when they're done being used, we're releasing them into the intermembrane space. And because of that, that's why we get this concentration gradient, where there's a lot more protons in the inner membrane space than there is in the matrix. So the mechanisms by which the proton gradient lead to ATP depends on ion channels that are going to be present, that are going to help that ATP synthase move, like I showed you before or discussed before. So basically, protons flow back into the matrix through channels in the F0 subunit, and the flow of those protons is accompanied by formation of ATP in the F1. Now, the mechanism for ATP formation isn't completely understood, but again, like I said, I have a video that I'm going to share with you that shows you kind of a model for how it works. Specifically, what we see with this coupling process is that electron transport causes pumping of hydrogen ions into the intermembrane space, and the ATP synthase enzyme allows pumping of those hydrogen ions back down its concentration gradient. And somehow it harnesses that energy on the F1 subunit to drive phosphorylation of ADP to make it ATP. What scientists have found is that there is some unique structure about the F1 subunit that allows for it to phosphorylate ADP to make ATP. So the proton gradient leads to changes in conformation in a number of proteins, including the ATP synthase. And if you look at this figure over here on the left, they've found that there are 3 sites for substrate on the ATP synthase molecule. And these sites interconvert as a result of the proton flow through the ATP synthase. So what you can see is that on one site, it binds to ADP and the inorganic phosphate, And on another site, it has a conformation that is binding to ATP. And then on another site, it's basically a leaving or release site. So there's these three different possible conformations. And this is what they think is caused by the proton flow that allows for making ADP bind to the phosphate and produce ATP. So again, let me encourage you to watch that animation video because me trying to describe it isn't amazing. But in summary, in chemiosmotic coupling, the proton gradient is the crux of the matter. The flow of protons through the pore in the ATP synthase drives ATP production. And in conformational coupling, a change in the shape of the ATP synthase releases the bound ATP that has already been formed. So we see both types of coupling occurring with our electron transport chain and ATP synthase. I want to talk to you about a couple shuttle mechanisms that scientists have studied. Shuttle mechanisms, basically, they transport metabolites between the mitochondria and the cytosol. So the first one is the glycerol phosphate shuttle. We know that glycolysis in the cytosol produces NADH. Most of our NADH is produced through the Krebs cycle. However, we do have some produced in the cytosol. And the problem is NADH does not cross the mitochondrial membrane, but glycerol phosphate and dihydroxyacetone do pass through that mechanism. So the glycerol phosphate shuttle makes 1.5 ATP molecules for each cytosolic NADH. So we know that we can make DHAP in glycolysis, and we also know that glycolysis produces NADH. The problem is NADH cannot be transported into the mitochondria in order to donate to the electron transport chain. So instead what happens is NADH donates electrons to DHAP to make glycerol phosphate. In that process, glycerol phosphate is able to be transported into the mitochondria, and then glycerol phosphate donates electrons to reduce FAD into FADH2. And we already know that FADH2 donates electrons to the electron transport chain, so therefore we can make 1.5 ATP molecules for every molecule of glycerol phosphate that's donated. And this is how the glycerol phosphate shuttle bypasses the fact that NADH cannot be brought into the mitochondria from the cytosol. An even more efficient shuttle called the malate aspartate shuttle has been found in mammalian kidneys, liver, and heart. So malate can cross the mitochondrial membrane, however, oxaloacetate cannot. And the deal is that the transfer of electrons from NADH in the cytosol is able to produce NADH in the mitochondria via this shuttle. And the nice part is that this shuttle allows for production of 2.5 ATP molecules for each cytosolic molecule of NADH. So how does this work? Basically, in glycolysis, you have NADH. And like I said before, NADH cannot get into the mitochondria. NADH that we use in the mitochondria is made in the mitochondria. However, we do have oxaloacetate outside of the mitochondria in the cytosol. And an enzyme called cytosolic malate dehydrogenase is able to reduce oxaloacetate to make malate. Malate can be transported into the mitochondria. So it's transported in the mitochondria, and there it donates electrons to NADH. NADH then donates electrons to the electron transport chain and is able to make 2.5 molecules of ATP for every molecule of NADH. At the same time, oxaloacetate is going to be regenerated because malate donated its electrons. Oxaloacetate is then converted into aspartate, and aspartate can also be transported out of the mitochondria. And then aspartate can be converted into oxaloacetate, and then the process can start all over again. So the idea here is that there are shuttle mechanisms that transfer electrons, not NADH, but electrons on other molecules from the cytosol across the mitochondrial membrane. In the malate aspartate shuttle, 2.5 molecules of ATP are produced for every molecule of cytosolic NADH, and there are 1.5 molecules of ATP produced in the glycerol phosphate shuttle, a point that affects the overall yield of ATP in these tissues. So in the end, how much ATP do we get from complete oxidation of glucose? Somewhere between 30 or 32 molecules of ATP. And that depends on which of those shuttle mechanisms are used in which tissues. Let's take a closer look at table 20.3 to get an idea of the yield of ATP molecules from glucose. If you look at glycolysis for both shuttle systems, you have -1, -1, +2, +2. So basically I have a net yield of 2 ATP molecules, and I have 2 NADH molecules produced in glycolysis. In pyruvate conversion to acetyl-CoA, I am producing 2 NADH molecules. In the citric acid cycle, we have 2 molecules of GTP that are produced that we're calling homologous to ATP molecules. So we have 2 more ATPs there, and we are producing 6 NADHs and 2 FADH2s. Finally, this is where it matters, oxidative phosphorylation. This is where it's all going to add up. So you're going to have 2 NADHs from glycolysis that are going to produce 3 ATPs via the glycerol phosphate shuttle and 5 via the malate phosphate. So you can see that's where our difference is because the yield is different. Oxidative decarboxylation of 2-pyruvate to 2-acetyl-CoA produces 5 for both pathways. 2 FADH2s produce 3 for both pathways, and 6 NADHs, which come from that citric acid cycle, produce 15. So overall, if a tissue type uses the glycerol phosphate shuttle system, they're going to produce 30 molecules of ATP. If a tissue uses the malate aspartate shuttle system, they're going to be able to produce 32 molecules of ATP for every glucose molecule that's oxidized. Okay, let's come back to our objectives and see if we've answered them all. You're supposed to be able to describe the purpose of the electron transport chain, check, and the complexes 1, 3, and 4 in particular, yep. ATP synthase, yep. Their substrates, products, cellular localization, and tissue distribution. Yeah, we talked about that today. Number 2, explain how electron transport chain and ATP synthase are functionally coupled. We talked about today, specifically ATP synthase coupling. Explain how the process of oxidative phosphorylation is influenced by the availability of oxygen and NADH. You know, I didn't talk about this, but I'm going to leave this here and ask you, why would oxidative phosphorylation be influenced by the availability of oxygen and NADH? Oxidative phosphorylation is the coupled process of the electron transport chain and phosphorylation of ADP to make ATP. So if I've removed oxygen, why wouldn't I be able to make ATP? ATP synthase seems to not require oxygen. Why would I not be able to make ATP? And the same thing goes for NADH. If I removed NADH, ATP synthase should be able to still work. It's not part of the electron transport chain. Why wouldn't it be able to work? So once you've watched this video, what I'm asking from you is that you email me the answers to both of those questions. That's it for today. I want you to know that the symposium report is going to be due on Friday. Basically, you just need to go in, watch your peers present their symposium presentations, and then evaluate them. And then our final Zoom meeting will be on Friday, where we're going to review for the final exam. All right, biochemists, happy studying.